Greetings, everyone. It's good to see you all again. On behalf of the Crop Trust and the CGIAR Gene Bank Initiative, thank you for joining today's Grow webinar. Before we begin, please note that comments and questions for the presenter can be placed in the Q&A box at the bottom of your screens. Should our Zoom connection fail, we will immediately receive a new email message. Please click on the link to join the webinar. It's my pleasure to introduce our speaker today, Dr. Nigel Maxted. He was born in Cornwall, UK, and has degrees in botany and zoology. His master's degree focused on breeding the tropical legume wingbean, and his PhD degree was on the temperate legume vetch. At the University of Birmingham, he ran the internationally renowned master's degree in conservation and use of plant genetic resources. His professional expertise is in plant genetic in situ and ex situ conservation, and on these topics, he has published over 350 peer-reviewed scientific articles, 220 edited book chapters, and 25 books on plant genetic resources. Most recently, his interest has focused on crop wild relatives and land conservation and the conservation policy development. He is currently International Scientific Advisor of the Alliance of Biodiversity International and CIAT, and Chair of the European Cooperative Program for Plant Genetic Resources, the Crop Wild Relative Working Group. The title of his presentation is How Can Gene Banks Double the Diversity Available for Plant Breeding? Welcome, Nigel.
Thank you, Yanni. That's a very kind introduction. And I'm very privileged to be here today to talk about this subject. It's really is a bonus for me to have an audience of gene bank managers listening to this talk, because it's designed very much for them. And I have a specific question I wish to ask them at the end. So thank you very much for the invitation to give the talk.
Welcome.
So shall we go to the first presentation? Thank you.
Yes.
Okay, so this is just a title slide. I just wanted to say on this that although I'm giving this presentation, the work included in this presentation is by literally hundreds of co-collaborators in different projects. So although I'm giving the talk, it's not only including my own personal work. A lot of it is based on, well, in fact, all of it is based on collaborative research. Next, please. Okay, so let's start off with a simple one. So this is a simple definition of genetic resources, the genetic material of animal and plants of value as a resource for present and future generations. We all know this. Why is genetic diversity, genetic resources important? Because of the changing ecosystem, either naturally or through climate change and changing consumer demands. So to continue to produce new varieties that are applicable for the time, we need to have continued access to genetic diversity. So this is contained within the crop gene pool, and the diversity in the crop gene pools is a key to future food security. Next, please. So if we look at the main sources, it says here in the bottom bullet point, 85% of conserved crop wild relative material is orthodox and held as seed in gene banks. So the primary focus of plant genetic resources and crop wild relative conservation is not in situ, but ex situ conservation. And so obviously gene banks are key to conserving this material. So what is the actual role of gene banks? So I looked for a definition of gene banks, and actually it's quite it's easy to get a definition of a gene bank, but not to establish what their roles are. So I've found this one definition back in 1991. So the role of gene banks is to maximize conservation, characterization, documentation, and use. The whole point is, if you're conserving the genetic diversity, it should be linked to use. And what you need, if you're doing the conservation element of this, and again, the use, the broadest use, is allowed by the broadest range of genetic diversity that's conserved. So the maximum number of alleles and gene combinations. So if we look at basically normal seed conservation via gene banks, which is outlined in this diagram here, then basically we have agrobiodiversity land races or crop wild relatives, either held in cultivation or in nature. It's located, sampled, and transferred to the gene bank, then goes through the process, the internal processes. Once entering the gene bank, is then stored. And if it's going to be used, it has to be characterized and evaluated and linked through, and the material made accessible for utilization by farmers and breeders. Next, please. So I'm going to focus in my talk really on talking about agrobiodiversity in cultivation and nature. And next. And then the link from the gene bank through into utilization. So next, please. So this is a diagram I'm sure you're all aware of, the 7.4 million accessions in the last FAO report, presumably 8 point something. Now I haven't seen the draft of the next State of the World report, but it's increasing, and you can see here the spread of where the gene banks are around the world, with the largest gene bank is Svalbard, managed by the Trust and collaborators. Next, please. Okay, so breeders, as I said, are the main end users of the genetic diversity we're conserving, but they themselves are saying there is a lack of sufficient diversity or access to diversity available to them to tackle climate change. And so there are various references stating the importance of increased accessibility to genetic resources. In terms of crop wild relatives, we know that they're suffering erosion extinction. So we need to do active conservation now, because if we leave it 10, 20 years, then that genetic diversity is not going to be available. So between 16 and 35, next slide, please. Or go back to the slide where it was. So if we look at the percentage of crop wild relatives that are conserved, it varies depending on individual studies. So some have greater estimates of genetic diversity being threatened within crop wild relatives than in others. But I would guess the 35% is quite a high figure. And I think the first one of 16% is more realistic for the percentage of crop wild relative which is actually being threatened currently. And the reason I believe in that lower figure is because crop wild relatives are often found in anthropogenic environments, so they're more likely to be in threatened environments to start with. So they're used to coping with those conditions. In terms of funding spent on conservation, 99% of crop wild relative conservation goes into ex situ. So in other words, the bulk of funding goes into crop wild relative conservation ex situ. And that material is now, as we can see, well by the Trust project, as well as Trust Crop Wild Relative project and the New Bold project are good examples of where that material is actually being used. So it does currently work. The relationship between ex situ and the breeder does currently work. But as we say, the breeders are asking for more diversity. And if we look at the actual holdings of crop wild relatives, then we can see about a third of them, a third of crop wild relatives have no material in gene banks, about a third are poorly conserved, so having less than 10 accessions per taxon in the gene bank, and 95%, as estimated by Nora Castaneda et al., require further additional collecting. But this all assumes that everything is based on ex situ conservation. And within conservation, there are basically two different strategies, in situ and ex situ. In situ including on farm. And in terms of crop wild relative conservation, today still in situ conservation is almost completely ignored. So there are very few active conservation of crop wild relatives within existing protected areas or other effective area-based conservation management. So that's outside of existing protected areas. And we also need to improve access to the crop wild relatives that are conserved within gene banks by improving documentation, informatics, characterization, evaluation, and then digitizing all this passport, characterization, evaluation data and making it available via the net. Next, please. Okay, so the fact that we need to take this broader approach is underlined in the policy documents in the latest draft of the post-2020 Global Biodiversity Framework. We can see milestone A3 talks about genetic diversity of wild and domesticated species need to be safeguarded, and at least 90% of the genetic diversity of all the elements of the gene pool conserved. 90%, that's quite a large percentage. And then the target 3, it specifically talks about conservation of species and genetic diversity in wild and domesticated species. And target 13 focuses on fair and equitable sharing of the benefits arising from that utilization. And it also is reinforced, we know now, by the UN Sustainability Development Goals. The first 3 goals are all closely associated with food security. Next slide, please. So basically what I'm arguing is we shouldn't put all our conservation eggs in one basket. We should be doing both ex situ and in situ, which is referred to as complementary conservation, if we want to have effective increased conservation. And I've given this photograph here is taken in Eskifoggia, a small seaside resort on the Turkish coast, has wonderful fish restaurants. I can highly recommend it. But this place is also, it has a small peninsula, and on that peninsula is absolutely jam-packed full of cereal crop wild relatives. So if we are looking for sites for in situ conservation, what we're looking for is sites that are full of multiple crop wild relatives. So we have more conservation bang for our buck by conserving a lot of crop wild relatives in the same location at the same time. Though actually this particular site is very close to the sea, as you can see, and so probably unlikely to be good in terms of climate change and long-term conservation. But we need to find sites like this elsewhere in more protected environments. Next, please. So this is a classic paper published by Tanksley and McCooch in 1997, where they showed that if we're trying to maximize conservation of genetic diversity, then we need to focus on the wild species, because the wild species are where most of the genetic diversity is found. A lot of diversity is lost through the domestication process between the wild species and the land races. And even more diversity is lost because in modern varieties, you're breeding for uniformity, so you can harvest everything at the same time, and they all have the same resistance and the same harvesting time. An interesting point that's often forgotten, I think, in this Tanksley and McCooch paper is they talked about 95% of genetic diversity in the gene pool of wild tomatoes is actually in the wild tomatoes, in crop wild relatives of tomato. So if we're looking at the goal is to conserve 90% of the genetic diversity according to the post-2020 indicators, then obviously crop wild relatives are going to be a major focus if we want to conserve that diversity. Next slide, please. So crop wild relatives, they're the wild ancestors of crops. They still have value because of their value as gene donors to the crop. You can transfer traits from the wild species to the crop itself. They have so much diversity because they haven't gone through the bottleneck of genetic bottleneck of domestication, as I said earlier. And we can broadly define them as all taxa in the same genus as the crop, or more precisely, those that are in gene pool 1, gene pool 2, and those that have been previously used or in taxon group 1 to 4. So what I'm trying to do in this diagram here is illustrate by the size of the circle different areas of the gene pool and how much genetic diversity they contain. So in gene pool 1, breeders' lines and current cultigens, relatively small amount of diversity, more in land races, but much more in gene pool 1b, the close wild relatives, the wild species, which are closely related to the crops, and the secondary gene pool, and then less diversity, usable diversity, in the gene pool 3, the other Hordeum species, as illustrated in this example for barley. So it does illustrate how much genetic diversity is located in crop wild relatives, which is what I wanted to stress. So if you're going to go for conserving maximum diversity, it's sensible to focus on crop wild relatives for conservation. Next slide, please. So our goal is in situ, because currently there is very little in situ at all. Maybe 4 or 5 genetic reserves, as originally formulated, exist around the world where you have conservation in protected areas, and crop wild relative conservation, and then linking that to sustainable use of that conserved resource. So the sort of thing we're talking about in setting up achieving this goal is to establish some kind of system or network for crop wild relative conservation globally, that would have backup ex situ, would be available for use. Now, it's important to make this link even from in situ through to use, and would then have benefits to all countries of the globe. So this system network is likely to contain localities that have multiple crop wild relative populations, and they're maintained at those localities according to agreed minimum standards, which we'll come back to later. The people that are actually managing those sites are custodians. These might be farmers if we're outside of a protected area, or if we're inside a protected area, it would be the protected area managers. Genetic resource centers, and here I've deliberately changed the wording from gene banks to genetic resource centers, because I think gene bank implies the link only to ex situ. If we're going to say that gene banks in the future will have a more active role also in situ, then I think we need to change the terminology, and the terminology would be more appropriate: genetic resource centers which grant access to in situ and ex situ conserved material. But these are important both for backup samples from the in situ, but also for the link through to user access. And I'll come back to this in detail later. And then other stakeholders. So here I'm thinking about other people working on biodiversity conservation, or breeders or farmers who want access to materials. So the network itself would be made up of organizations, networks, individuals that would all form part of this greater network. And I also should point out at this stage, so a lot of this information I'm giving has come out of the developments in the Farmers' Pride project, which you can see the logo of up here in the right-hand side. And that's a project that recently finished and has produced a lot of information for going forward with network development. Next slide, please. Okay, so why is it important to focus on in situ conservation of crop wild relatives now? Because breeders are already saying they're unable to totally deal with climate change unless they have greater access to diversity. So we must meet those breeders' needs. That crop wild relatives, as I've already said, are a threatened resource. They're threatened globally, regionally, nationally, not just in their hotspots. So we need to do better conservation, more thorough conservation. There are existing gaps, both in conservation gaps in situ. Virtually nothing is conserved in situ, so a big gap there. But also, as we heard earlier from Nora Castaneda's work, obviously gaps also in terms of ex situ conservation, and then availability of that material. So not only is the material not conserved, but it's also, if it's not conserved, it can't be available, but some material that is conserved is also not available. So we need to increase the availability of that material. And we need to improve the complementarity in terms of global, regional, and national level conservation. They need to be a greater integration between the different levels if we are going to meet these legislative obligations and conserve crop wild relatives and use them for future, guaranteeing future food security. Next slide, please. Right, so I wanted to go through a bit of the science associated, or the GIS analysis really in this case, associated with global crop wild relative project development. Okay, so first, next please. So this is a paper again from, or one we're just showing, from Holly Vincent, 2019, and she did taxon richness for global crop wild relatives, looking at 1,261 crop wild relatives. related to 167 crops. And you can see here, it very nicely shows areas of the world that we would have expect to have high levels of crop wild relative diversity in the Mediterranean, the Fertile Crescent, but also areas outside of the traditional Vavilov centers. So look at North America there. Both the Western and the East Coast have crop wild relative diversity not noted by Vavilov, and then areas of, say, in Africa, West Africa, coastal West Africa, around the Gulf of Guinea, and Northern Australia, for instance, all showing additional areas where crop wild relative diversity is present. Next slide, please. So this is from Nora's work, again showing the ex situ sites where we need to focus our future ex situ collecting for ex situ conservation. Again, showing eastern seaboard of North America, areas in South America, the Mediterranean. Surprisingly, one would think that would be an area that would be well conserved, but surprisingly does come out as an area who benefit from further conservation. And then in Southeast Asia and Northern Australia. Next slide, please. So complementary to those ex situ goals, we have the in situ goals. So here we have the top 170 sites around the world where we need to set up, where we know there is a concentration of crop wild relatives. Each species, including these 170 sites, each species has 5 sites they're located in. They were chosen not arbitrarily, but they were chosen to maximize the range of genetic diversity within that species or taxon. And then all sites have been climate change proof. So in other words, they will exist for a longer time than other sites that might be destroyed by climate change. So these 170 sites are indicative of sites that would be included in any global network of in situ conservation sites. Next slide, please. So this is basically a revision of the Vavilov centers, adding in the additional centers identified by Nora and Holly Vincent's work, showing where the additional centers to the traditional Vavilov centers, as indicated here in fawn, and the green ones indicate the additional centers to be added. Next slide, please. So this is what I've tried to do here is start thinking about, if we're thinking about in situ conservation and the relationship with protected areas, is can we focus just in global hotspots, global biodiversity hotspots? Would they then provide good crop wild relative conservation? So you can see here the brown areas are the areas that are crop wild relative hotspots, and the blue areas are the Myers or Mittermeier hotspots of biodiversity. And you can see there isn't a complete overlap between these. Actually, there's quite a large disparity between biodiversity hotspots and crop wild relative hotspots. Next slide, please. And this is one I think is very important at the moment. So here what we've done is, those of you who know about the work of IUCN will know that they're taking forward their key biodiversity area concepts globally, and they're working out globally where are the best areas for biodiversity conservation so that biodiversity conservation activity can be focused in these areas. So the blue dots are crop wild relative occurrence in key biodiversity areas, and the red dots are found outside of key biodiversity areas. And very interestingly, if you look at the percentage found inside key biodiversity areas and outside, next slide, then you'll see that 25%, only 25% of these crop wild relatives for sub-Saharan Africa are found in key biodiversity areas. 75% of crop wild relatives are found outside of IUCN key biodiversity areas. Now, this is a shame, because what we can't allow to happen is that going forward, that key biodiversity areas are said to be holding representative material of crop wildlife. They do, in the sense that they do contain this 25%, and we can conserve those 25%. But in other words, the 75% is such a high proportion that are not found in key biodiversity areas. We also need to make sure we have in situ conservation outside of traditional biodiversity context. So we need to not do it ourselves. We can't rely on other organizations to conserve these species for us in situ. Next slide, please. Okay, so why do we think a network is, we could have a lot of standalone in situ sites. Why form them into a network? Well, if you have a network, then you can have your systematic coordination and reporting. You can form stronger partnerships. You can make sure, as I argued earlier, that you have integration between the global, regional, and national actions. You can work more precisely with local communities in developing common goals. And you can facilitate access and benefit sharing. So basically, if you ask any protected area manager how they can guarantee, if they provide crop wild relative material to a user, they will have their rights protected through the normal ABS conditions that are available to everyone else, they wouldn't have a clue. So basically, if they're working with a network, we can help protect these rights and ensure that farmers and protected area managers can have their full facilitation of access and benefit sharing, aided by the networking process. We can ensure complementary in situ ex situ by the backup that would normally be provided from the in situ to the gene bank. And we can also promote access of plant genetic resources via the gene bank. And I'll come back to explaining this in a later slide. And also, if we're doing obviously in situ, we have involving populations that can be secured for perpetuity. Next slide, please. So this is a photograph of Paul Watkins. He's probably one, if not the person in the UK who's most responsible for landrace conservation. He has 4 or 5 land races on his farm of different crops. And he's very dedicated to conserving land races. But also on his land, he has crop wild relatives found around the margin. So why should he be interested in joining a crop wild relative conservation network? Because of the kudos, prestige of being involved in having legislative protection of his site, as would come through setting up, being a member of an established network, contribution to bigger, stronger partnerships, assistance in adding value, developing markets for his products, sustainable use, technical support training in terms of maintaining his crop wild relative populations, gaining subsidies. In the UK, we're lucky enough to have a new agriculture bill that will actually pay farmers to maintain crop wild relative populations on their land. So that again, because he is a known person having a dedication to landrace conservation and he has crop wild relatives, he will definitely receive payments from the government and win out as part of this process. And we can make sure his material is securely backed up. So if for some reason he loses a particular accession, he can have it repatriated. Assistance with his access and benefit sharing, as I've argued earlier. Access to greater breadth of genetic diversity if it requires it, and then safeguarding, coordination, and other activities. So there are real definitive benefits coming to Paul from joining a network. Next slide, please. So these are various tools we produced in Farmers Pride, which I think will be very useful in terms of people, in terms of in situ, but also I think probably ex situ conservation as well. So conservation planning, we've helped to extend CAP Phytogen, which is a group of tools funded originally by FAO, but has now been extended as a result of additional funding we provided to them. We've developed the concept of in situ conservation, crop wild relative maintenance and availability access via EURISCO. We developed a group of descriptors for those. We developed a tool for looking up protected areas in Europe and finding which crop wild relatives will be present in each protected area, particularly in national nature reserves and Euro sites, Natura 2000 sites, and then population management guidelines. We've also developed detailed population management guidelines for how to manage crop wild relatives in situ. Next slide, please. So in terms of in situ, we have, as I said earlier, two kinds of sites. We have protected areas and we have these other effective area-based conservation measures. And both of them can be used. I stress both of them can be used for crop wild relative conservation. So originally we focused on crop wild relative conservation in protected areas simply because they have a long-term conservation ethos. There won't be any hasty management changes, so we're more likely to sustain crop wild relative populations in the long term. It's relatively easy to change the management plan of a site and include specific actions for managing crop wild relative populations. The cost is not prohibitive because you don't have to buy new sites to conserve crop wild relatives. And the existing protected areas, as we heard earlier, do have large numbers of crop wild relatives within them. But increasingly, we realize that with crop wild relatives, they are found in often disturbed anthropogenic environments. So we also need to look at conservation outside of protected areas. And so several years ago, we came up with these extra protected area-based conservation. But the IUCN more recently have caught up, I'm glad to say, and come up with their own name for these areas outside of formal protected areas. And OECMs are important because, as I said, a lot of crop wild relatives are found outside protected areas. They're less formal, so you would have to have legally binding agreements with the landowner or farmer, but they make a very good complementary measure towards conserving crop wild relatives in situ and protected areas. So we need to do both, basically, is what I'm saying. Next slide, please. So this is just stressing the obvious and perhaps trivial point that you have hundreds of individual crop wild relative populations, and some of those populations will form part of the national network of crop wild relatives. Some of those will form part of the European network, and some of those will form part of the global network. So the point being that you do have to have integration of national, regional, and global conservation in situ measures to have a good working network structure. The next slide, please. Okay, so this is just showing how we select sites to be included in this case, in a regional structure. So the green sites on the left-hand side of this diagram show the national process that goes into establishing crop wild relative, a national strategy for establishing a network in a particular country. So you start off with a list of your crop wild relatives. Usually it's quite a long list, so you would want perhaps to prioritize it, and you may prioritize it on threat, relative threat, or how important, how close it is to the crop itself, so how easily it is to transfer the traits, or whether it's an endemic species or some other estimation of value. You then come up with your list of definitive crop wild priority crop wild relatives, and then you do your gap analysis. Your gap analysis tells you where your coverage of maximum diversity is located. So which combination of sites gives you maximum diversity, and then you can implement that, and that becomes part of your national strategy. So that national strategy, you would identify also some sites which you think are perhaps your top 10 best sites in the country, and they would go on to become not only national sites, but also regional sites. So that would then feed through in the nomination of the most appropriate wild plant populations, and that would then go into a contribution to the European network, But you would also repeat the same exercise at the European level. So that's what you see on the right-hand side here with the blue image. Basically, you have the same process, but at a European level. So you have both a top-down and a bottom-up approach to building a European or regional network. And I think this is the way we're tackling it in terms of establishing the network in Europe anyway. Next slide. Right, now I want to take some time and explain this properly. So sorry, it's quite a complicated diagram, but I think you'll see it's not too complicated. Okay, so here on the left-hand side, we have the in situ process. And as part of that process, we would expect always to have some backup into ex situ. So as a routine part, even if we do nothing else, we would have a backup to the ex situ side. But also we need to make sure we can provide the link between in situ conservation and use. Very important. It has, if in situ is ever going to be as valued as ex situ, we must have that link between in situ and use. So how do we actually provide use to the material that's held ex situ? So the first option would be to actually go to the protected area manager, go to the farmer, and negotiate with them access to the material, sign your material transfer agreement, and then do whatever is appropriate to assure access and benefit sharing. But the problem is that most protected area managers know nothing about this. Remember, their biodiversity, their focus is on biodiversity conservation, not specifically crop wild relatives. So we can't expect them to engage in this process. And frankly, 99% of them, in my experience, don't want to engage in this process. So we need to think of some way that doesn't involve actually going to the protected area manager and asking for the material. So one way of providing access is through ex situ. So the protected area manager was saying, yes, I have no obligation, no objection, providing I have nothing to do, actively do in terms of promoting the access and benefit sharing. I will allow my material to be used, providing I get some access and benefit sharing back to me, but I don't want to actually go through that process myself. So it would actually be done by the gene bank. So the material would go from the in situ resource to the gene bank, and then would, as the gene banks are already experts in providing material to users, the resource would be given to the user. User would pay the any benefit back to the gene bank, and that will go back to the protected area manager. So you have the flow of resources in that sense. So within that context, then we have 4 different options. So the first option is to do exactly the same as we do now for any long-term ex situ material. In other words, the material passes into the gene bank, goes through the normal cycling process, and importantly goes through regeneration and everything else. Now that, when immediately you say that to gene bank managers, they say, oh, it's enormous. There's going to be a huge additional expense associated with this, quite rightly so. And so they frown on that as an activity, or in my experience, I've had lots of feedback that they do so. The second option is to have essentially a black box. So you send the ex situ material to the gene bank. It's registered, clean, dried, germination testing done, and then put in a black box in the gene bank. We all know about black box, but the reason we don't have routine ex situ black box storage is because it doesn't then provide access to users. So there is no link, this intimate link we're trying to create with users. So black box is not an ideal option, in my view, for in situ material. So you could have in situ backup and supply. And what I mean by this is the material passes from the in situ source, is registered, documented, clean, dried. Perhaps you do germination testing, but you don't have to. And then packaged and maintained within the gene bank, but not regenerated. And so that's why I say you don't need to do the germination testing, because if it's not regenerated, maybe you don't need to do the germination testing either. And how you get round the loss of viability over time is you just periodically do resampling from the in situ source. So that replaces the expensive option of regeneration. A fourth option would be demand and supply. So you advertise what material is available from the in situ resource, the user sees it, and when they ask for something, you supply it. Now, the slight drawback of demand and supply is that it obviously takes time, and due to seasonality, you can only access seed at a certain time of year for a limited period. And so there may be actually quite a disbenefit of having a demand and supply system. So those are the four options. Now, my personal view, and remember I'm in this case arguing for in situ, the best option for in situ I think is option 3, this backup and supply, which doesn't require so much work by the gene bank, but does still deliberately make the link between the in situ resource and the user community, access to the user resource, as you can see here, as identified in this diagram. So I spent a bit more time on that because I think it's important for you to understand what I envisage might be taken on by the gene bank as an additional activity. Next slide, please. Okay, so coming to the end now, I want to talk a little bit about governance. So governance, as defined here, is the rules, norms, actions are structured, sustaining, regulating, held accountable. So how is the network going to be effectively managed? How's it going to be sustained in the long run? And that's where we ran into a difficulty in the Farmers' Pride project. We didn't quite get there. We didn't quite identify the European body that would take over the actual management of the network once established. But there are various options you can see here in Europe, ECPGR, the European Environment Agency, European Commission itself, I think, is probably the most likely organization to take on this role long term, but or Europark or Eurosite, but we don't know that this time. In terms of national networks and who would take over the governance structure, well, the national PGR agency is the obvious target for that, but in some countries, maybe we would have a protected area community who are so into doing in situ conservation crop wild relatives, they might lead this activity themselves. And globally, there are obviously various options there for providing the governance structure. But these are the sort of issues I think we really should be discussing openly at the moment. Who's going to take the lead in these actions? But the important thing to realize when we're talking about governance is still the resource has national sovereignty over the control over the genetic resource. So all sites would have to be nominated for in situ inclusion in the national, regional, global network by the national PGRFA coordinator. All sites are under the jurisdiction of national agencies, and access would have to have some kind of control via the national authorities. Next slide, please. So in terms of, I mentioned earlier about the management of the network, management of individual sites. So we have here a set of criteria that we've put together that might be involved in laying down the minimum criteria a site would have to meet to be included, or the populations within the site would have to meet to be included within the network. I won't go through them all in detail, but you can see the kind of things we're talking about: native, there's routine sampling for ex situ backup, etc. And it's a healthy population, not threatened. Next, please. And this is for individual sites, the kind of criteria you would have to meet to have passed the minimum standard for management of the individual sites. And again, I won't go through these in detail now. Next slide, please. Next slide, please. Okay, so this just shows the process of adding sites to the network. So you have basically a site which you think has important, is a hotspot for crop wild relative diversity. You review the characteristics of that site against the characteristics, the criteria for entry into the network. You then nominate it. The nomination comes from the national PGRFA coordinator, goes to the management committee of the network. They review it, see if it meets, the site meets the management criteria. If it does, then it goes into the network. And then if it goes into a regional network, in this case, Europe, but if it goes into Europe, then it potentially could also go into the global network and be conserved both at national, European and global levels. But obviously it wouldn't be put in forever. So you'd have to have periodic reviews to see that it still maintained met the criteria for being included within the network. So if it passed those criteria, it's maintained in the network. If it fails, then it would have to go back and be re-nominated by the national government. Next slide, please. Okay, so I said that we didn't get quite to actually establishing the European network, the final network within the Farmers' Pride project. But what we did do is we came up with a self-notification system where if you were managing crop wild relative or landrace diversity in Europe, you could nominate for joining the network once it was established. And perhaps even I was surprised at the number of people who were keen to nominate their sites from throughout Europe. And this is just really in relation to one project. So I emphasize that I think this is quite a good indication that the time is right for setting up in situ networking at this moment. We're ready to do it. Next slide, please. And I wanted to show a slide to draw attention to setting up a network in a different region. We've also been working with Biodiversity International, notably Dr. Aishan Dalou, on establishing a network in the SADC region of Africa. And there we did manage to set up the governance structure. So in that case, we went through various stages of producing a white paper, putting that before technical committee of the SADC region and the crop directors, and then finally getting it agreed by the SADC joint meeting of the ministers of agriculture in the region to actually set up an in situ crop wild relative network, because they saw the importance of making crop wild relative diversity available for a wider audience. So I've just added here that Hawkes in a paper in 1991 said that in situ was in its infancy. And I hope you've heard from what I presented today that in situ is beyond, well beyond its infancy. We really are at the stage where, rather than just having me rambling on about in situ conservation all the time, we can actually really start setting up in situ networks nationally, regionally, and globally. Last slide, please. Okay, so this is my last slide. As I said, PGR centers, remember, not gene banks, plant genetic resource centers, I believe, need to become active in terms of in situ conservation, both engaging in situ planning, but also engaging in setting up in situ conservation and making that link through to utilization via national gene banks, CG gene banks, other gene banks. Now, I'm sure the gene bank managers among you are, sitting there thinking, God, he has no idea of the real cost of this. I think it's actually the reverse. It's the real benefit of doing this in terms of, I've put here in quotation marks, doubling the diversity available to breeders. I have no idea, basically, the percentage of benefit from making ex situ as well as in situ material available to users, but I'm sure it's going to, frankly, more than double the amount of diversity that is currently available. And it's an action that we can't sit on our hands with 850 million people today suffering malnourishment. Those people need us to take real steps into the future. And that step involves going forward with making this link between in situ conservation and crop-wide relatives and the user community and gene banks. And so lastly, next point. My last point is, as I'm talking, I hope, to a lot of gene bank managers, or should I say plant genetic resource center managers, what do you see as your role? I hope you do see, or are beginning to see, or encouraged to move towards in situ conservation as well as ex situ. Thank you very much for listening to my presentation, and I look forward to answering your questions.
Thank you, Nigel. That was wonderful. There's a lot there to digest, and this is evident in the number of questions that we have. So I'd like to start with Dr. Siddiqui's question. He says, how to fill in the gap, particularly for those developing countries, for pre-breeding and technology and use of crop-wide relatives in crop improvement. So his question is, how do you fill in that gap that we currently have between the pre-breeding and crop management? How, in your mind, how do we start?
Well, I would have thought we have already started. I mean, that's what I shouldn't be telling him. I thought he was involved in the Trust Bold project, but the Trust Bold project is all about filling this gap. It's about working, the CG gene banks, working with developing countries to provide pre-bred material that national breeders can use in their breeding programs. So I think we're actively working towards it already. The thing that I am perhaps a little discouraged about is, well, one, that we're not effectively doing in situ conservation of that material. So the pre-breeders can't do pre-breeding if they don't have access to the crop-wide relative diversity. So what we do need to do is make sure we have much better conservation of crop-wide relatives, so the pre-breeders can actually have access to the maximum range of diversity to use in their breeding programs. So that's basically how I would answer the question.
Okay. Does the term KBA refer to only plants? And is it similar to the term IPA mentioned in the Global Strategy for Plant Conservation?
So it doesn't refer just to plants, it refers to plants and animals. So it's a biodiversity. IUCN works on biodiversity conservation. No, it doesn't. It covers both. Okay.
So from Edwin Cockman, are there any countries or regions that are particularly good at in situ conservation of crop-wide relatives and a network that we should perhaps imitate elsewhere?
Right, so that's a bloody good question. So I hate to say this because it sounds as if I'm blowing my own trumpet, but I'm European. And so we have had a series of projects in Europe working on this. And our last project, as I said in my talk, was Farmers' Pride. And I deliberately, when I was writing the project document, the application, put in one of the deliverables being a self-sustaining network at the end of the project. And as I reluctantly admitted when I was giving my talk, we didn't get there. And we didn't get there because we didn't sort out this governance structure. But I would say that, frankly, that Europe is more advanced. Now, I did also mention the SADC region, and the SADC region, we do have the governance structure there, but I would say on the ground, the actual network itself has not been actually established. So although they have the governance structure ready, it has not formally been established. And I know that Aishan has applied for more funding to set up the formal structure within the network once it's developed. So I think Europe, but if you and I have to admit to you that we don't currently have a network. So there isn't a network structure you can follow, but I think the most progress has been made in Europe, and certainly a lot of the methodologies of how to do it we've established in Europe already. So sorry, not a perfect answer. I wish I could be more positive.
That's good enough, I think. We go back more to a theoretical question. Can you please tell us about genome plasticity in changing climate? And I think it refers to your diagram where you say that you resample the place, if viability goes down within the gene bank, that you resample the place where the crop out relative was. What happens if this sample was taken years before? There have been floods, drought, etc. So there is, some change in that original, due to climate change, there was change, genomic change in that crop out relative. What do you do then? You're sampling basically a different set of crop out relatives.
No, you are. But remember, there is a difference between in situ and ex situ conservation. In terms of ex situ conservation, you're taking a sample and you're holding it forever. In terms of in situ conservation, remember the whole basis is it's not held forever. It continues to evolve. And so what you're doing is you're sampling the in situ population at any one time. If you then put it into GeneBank and treat it as a normal ex situ sample, then you've not got a problem with your plasticity. But if it's, as I was arguing, it's a replacement for in situ conservation, it's making access to the in situ material, then it doesn't matter that you have a change in the material over time. You would expect that. If you want to have permanence, then go for ex situ. But if you want to have dynamic, then go for in situ. And that you should be going for both. I'm not arguing for one over the other. And I'm certainly not arguing, when I beg for money for in situ, I'm certainly not arguing that we should cut money for ex situ, because obviously ex situ currently works. That would be extremely foolish to take money away from ex situ and put it into in situ. But I am arguing that we need more diversity, and that more diversity can only be available if we have in situ and ex situ conservation.
Yeah. So basically you're arguing to have a branch within the Plant Genetics Center that deals directly with in situ conservation and how to bring those genetic resources within the bank. And I think
make them available. Remember,
the
really important thing is we have to link through to availability. Otherwise, it becomes like when I went through that diagram, I was explaining the option of having black box. So black box doesn't work for me because it doesn't provide any access to the in situ material.
Exactly. Yeah, they would have to have availability. I think several of the plant genetic resources or centers that we have visited do have that. They do have a link to the farmers and a link to the in situ. However, it's not an established work plan for them to do that. And I think that that's where one of the gaps are. Some of the new projects now have that as well. So I think that link to in situ must be created more formally, I believe.
Yep.
Okay, so we have
I completely agree. That's what I was asking for or begging for.
Right. From Tinatin, how is it possible institutionally to link ex situ conservation in gene banks with conservation in situ? Any stories of success?
Well, I think we're starting to see stories of success, for instance, in my own country, in the UK. We have the government is actually funding development of the network, the in situ network, as we talk today. I mean, they're providing funding for making clearer links between UK gene banks and the in situ resource and working with protected areas in the UK. Now, I don't think that's the only country in Europe where it's happening. In Germany, I know it's also happening. And I think also in the Scandinavian countries and probably in other countries as well. So I think we've been talking about this for so long that it just takes time for the message to get through, unfortunately. I think it is now is the time, basically. And you just said yourself that in situ was going to be included in the CG's remit within the gene banks. You talked about them being more involved in situ efforts. And yes, I realize that myself. And I think that's why we've come to the time where, and the methodologies there, as I tried to describe, we have very detailed methodologies now on how the criteria for acceptance of sites into the network, how the network would be run, et cetera, how crop wide relatives will be managed in situ. All of that information is now available. Now, at the moment, it's going to remain theoretical unless we start applying it. And once we start applying it, my guess is they will change. Some bits will work, some bits won't work. But we're at the stage now where we have to start applying these methodologies to see how they work and how they develop in the future.
And related to that, from Milona, it would be nice to have some examples of crop wild relatives in situ conservation and their exchange. And have there been benefit sharings? Do you know of instances where there have been benefit sharings once the crop wild relatives have been used in some of the products?
No, I really wish I could give you specific examples, but I can't. And I'm talking about the in situ context, of course, not ex situ. But I can't give you any examples yet. We just aren't at that stage. I mean, how many sites do you think around the world are doing active in situ conservation of crop wild relatives at the moment? My guess is it's still, if you look at the criteria we've established on how to select sites and how to manage the sites, the number of sites that are actually doing that already would probably be less than 4 or 5 globally. And they've only just been established if they have. So we haven't got to that stage yet, but we'll never get to that stage. And yes, unless we make this link now between in situ, ex situ through to use.
And an added complication that I see, Nigel, is that most of the gene banks fall under the Ministry of Agriculture for national gene banks, whereas the protected areas for crop wild relatives, for example, they fall under the Ministry of the Environment. And so you're actually talking to 2 different ministries to try and connect within the same country, and that can be a huge challenge.
Yes,
for them to.
You're telling me I've been doing this for the last 30 years. I mean, I mean, yes, I know. I bear the scars of that activity. But look, I mean, this, joking aside,
yeah,
I think this is so important now. We're not, you know, I wasn't meaning to jest when I said at the end about the 850 people who are suffering malnourishment, 850 million people today who are suffering malnourishment. If we are going to feed them, we need to do everything we can to improve food security. And one clear way of doing that is to have greater access to diversity. And I don't see, even if you spent billions on further ex situ collecting, I don't see that providing a doubling of the amount of diversity available to breeders. This, I can tell you, is going to more than double the diversity that's available for breeders if we can set up a system that works.
Okay, here is another one from Yoko. In the in situ ex situ link, how do you calculate the time scale of the access and benefit sharing to the in situ germplasm provider? Generally, it takes decades to produce a new crop variety, and the original beneficiary who signed the MTA might not be there. Long-term involvement for need to be guaranteed. Long-term involvement is to be guaranteed. So what happens if the original beneficiary is no longer there? Access and benefit sharing after 1 decade. How do you see that?
Not a question for in situ versus ex situ. This is a question for genetic resources, full stop, because it applies both to both. Yes. And you could argue that, I mean, I don't want to argue this, but I'm going to put it forward, is if you have in situ, then there is a long-term commitment, certainly in situ in a protected area, then there is a long-term conservation commitment to that site already. And it will go on for presumably into the future. Very few protected areas become unprotected and just go back to being normal land. So in those cases, you do have the long-term beneficiaries potential. In other words, they're going to be there for the next 100 years. So there is the potential for the material to come back to those users. If we're talking about individual farmers, then the individual farmer would be exactly the same as an individual farmer who had given ex situ material. It would be exactly there's no difference in the relationship. Though I would want to make sure you had a permanent agreement. You know, you have to have a written agreement with the farmer if they're managing in situ crop wild relatives on their land. And I would hope that that went on from generation to generation within the farm if it was passing down the generations. But I don't, yes, I suppose really what I'm saying is I don't see any difference between in situ and ex situ as regard that question. Okay.
Here's a matter of giving priority. So in case of limited ex situ facilities, in other words, the gene bank is small, which of these crops need to be prioritized? Crop wild relatives with unknown potential traits or improve varieties which contain some desirable superior traits? And there's a second part to this question.
Well, so the answer is I think it's a false distinction. So are you saying the gene bank would provide you know, sort of breeders' lines? And I mean, the breeders' lines will be there anyway. I think that it's not a distinction between conserving or working on in situ conservation of crop wild relatives or working on breeders' lines. I think the breeders' lines will be there, and I think, you know, if the money isn't there, then you don't do the crop wild relatives. I guess it's as simple as that.
Okay.
So the next part of your question?
Crop wild relatives are usually less economical. How to engage farmers to participate for its conservation?
Well, I think they need to understand what crop wild relatives are. I mean, one of the things I do, and I guess all scientists do this, we occasionally give talks to local, certainly I do, to local farmers on what I'm doing. And when you go up to them and you know, initially you arrive in the hall and you say, I work on crop wild relatives, and they think, oh God, another scientist, what's he going to talk about? Some theoretical subject, etc., etc. But immediately you give your talk and you explain what crop wild relatives are, they see it, it clicks in their head. You can see their view changes. They start to smile because they see the direct benefit of maintaining a resource for their children, because most farms are passed on, you know, father to son, mother to daughter. And so they want a long-term resource that can be passed down the generation. And this secures that resource, because then you can have, like the biodiversity have various projects, the Seeds for Needs projects, for instance, where they provide some pre-bred material to farmers to cross with their own land races to maintain the land races and give them some climate change immunity. So that's using crop wild relatives and the pre-bred material, crossing with land races, providing long-term security. So I think they get it. I think initially, if you talk to people about crop wild relatives, they don't get it. But once it's explained clearly to them, the benefit, the long-term benefit, it's not rocket science. It's actually quite simple. And it's the sort of stuff that they've been doing as farmers for generations.
Yes. Okay. There's one here about land races, which might be interesting, and I'm hoping I can interpret it. I think developing countries are yet to utilize genetic diversity available in land races. Under such situations, can we convince policymakers, or how can we convince policymakers to invest in utilizing this type of germplasm?
Right.
In other words, what would be the direct steps that a gene bank or an institution would have to convince policymakers to set aside land, to protect these lands, work crop relatives, and also protect land races and other different types of genetic resources?
Right. The land already exists. So if we're talking about specifically crop wild relatives, then, for instance, in the UK, how is the government going to fund conservation of crop wild relatives outside of protected areas in the UK? Well, how they're going to do it is they're going to fund farmers which already have crop wild relative, healthy crop wild relative populations on their land. They're not going to give money to farmers that don't have crop wild relatives. So the populations are already there. So the farmer doesn't have to do anything different. It just has to basically maintain the management of the land that is currently, the way it's currently managed is maintained in the long term. And then they will get this reward to support them for doing it, to acknowledge the public good they're doing by maintaining these populations. So if you compare that with maintenance of land races, I think that both are important, frankly. And the argument, if you force me to come up with an argument for benefit of one over the other, I would say with crop wild relatives, as I argued in my talk, they have greater diversity because they haven't gone through the bottleneck of domestication. Yeah. But I don't like to be put in the position of favoring one over the other. Both are important and both should be pushed forward by the government, by the national gene banks, etc.
Wonderful. We're getting close to the time when we should slow down a little bit, maybe even say goodbye, but there's a couple more good questions. And if you're in for it, Nigel, I'd like to
I'm still raring to go.
Okay, wonderful. Lazardidi would like to ask, how often should a gene bank resupply in situ sites? Under which criteria, and what about the population numbers? So how often should, if there is a branch from plant genetic centers, how often should they be going out to these excursions and resupplying, in your mind?
Right. I think they shouldn't be going out at all. So basically, when you make the agreement with a site manager, whether it's a farmer or with a protected area manager, part of the agreement is hopefully, like in the UK, they receive some kind of payment for being involved in the network. And if they do, then part of that payment is they have to provide regular samples, and the regular samples to go from the site to the gene bank as the backup, and then as a basis for utilization would be for an annual species. I think if you did it once every 10 years. But then if you've got a particular accession, say you're a farmer on the edge of a desert, and obviously your material has drought potential, then maybe users are going to be more interested in that. And so as the material is used up from the gene bank, and remember, you can use all the material up, you don't have to keep some back for regeneration. Then as soon as it's used up, you can get more from the in situ source. But my guess is in terms of significant change in the population, if it's not being used by users, then I would suggest for an annual species a backup of once every 10 years. And then obviously, if you're dealing with a perennial fruit tree, then it wouldn't need to be anywhere near so often. So it would vary depending on the lifespan of the species.
Yeah. Also, living in an area that is prone to very high drought, very cold temperatures in the winter, and flooding, extensive flooding, 2 to 3 months of flooding, I would say if there have been extreme circumstances in that land, it would be good after 1 year to see what it was that came up and resample after those events. Because as I can see in where I live, I mean, there's a different flora now, as there was 5 years ago before we had the deep freeze and before we had the hurricane. So I think one of the things we have to do is resample once climate is forcing us to do so at every event. And I think some interesting things have come up.
Yeah, I agree. But I think these sort of details, frankly, they are details, and they would need to be sorted out once you've got the network up and running.
Right.
Because the important step is to actually stop talking about it, start doing. Set up the network now, and then we can discuss and we can get people who really know what they're talking about, rather than me just blathering on.
Okay, here's an interesting question from Nicola Rowe. Ex situ gene banks are organized according to crop types, for example, clonals, cereals, forages. Would you do the same for in situ conservation?
Well, yes, I guess so. I mean, I suppose if the material is not suitable for seed storage, then you would have no alternative. You'd have to do that. But I must admit, most of my thinking so far, and the actual practical work I did as a plant collector myself, was always as seed collecting. I never did any other kind of collecting, and so maybe my thinking is a bit narrow on this. But I mean, yes, I do think that would, if you have material which is as standard is collected as cuttings, for instance, then you would have to apply the same technique.
Okay. A more general question: if we have the crop wild relative, what kind of research should we start in for pre-breeding?
What kind of research? Well, I mean, I suppose I hate to suggest this because it's not my area. I have to be honest, I am a conservationist, but I mean, I guess something that I always thought is gene editing is going to be something that is really advantageous for working with crop wild relatives. Because the big problem, if you talk to breeders, the breeders I speak to will always tell me, well, crop wild relatives, they've got all that linkage drag and all that rubbish we get with the traits we actually want. So in terms of genetic engineering and gene editing, it does give you a chance to get the genes you want specifically, rather than having all the background. And so that, I would think, is, if we can find an easy way of doing that, then that would be advantageous. The only reason I slightly hesitate in suggesting that is because that's a tool that is available in the best labs in the world. It's not a tool that's available in most developing countries for the average plant breeding program. So we have to be realistic in the kind of things we're suggesting. And in that case, I guess we're talking about more standard characterization evaluation, actually finding these adaptive traits that can be which accessions have the material that's important to be used in the crossing.
Thank you. I think with that, we'll end. There are a few congratulations, very informative. Thank you very much. I should let you know that the chat is filled with these very nice compliments, Nigel. Thank you very much for a very interesting presentation. To all of you, thank you for your attendance. We wish you happy holidays and hope to see you back in February on our next Grow webinar. Any last words, Nigel?
Well, like you, I'd like to wish everyone happy holidays and Christmas and New Year. And thank you for listening. And I hope that I've been able to draw a bit closer to the establishment of national, regional and global networks for crop bioderivative in situ conservation. Thank you very much.
Thank you to all. Bye-bye.
Bye.